I want you to think about the last time you saw a news ticker scrolling across the bottom of your screen.
Oh, yeah. Or maybe like a lab report from your doctor.
Exactly. Or
Ugh.
even just reading a warning label on some recalled spinach?
Right. You see those names all the time.
Yeah, you probably saw a name flash by like E. coli or C. diff. And, well, when we see those names, we generally just accept them.
We do. We treat them like absolute facts.
Right. We accept these scientific names as absolute permanent facts of nature, like they were, I don't know, carved into stone at the dawn of time.
It carries this immense weight of objective authority, doesn't it?
It really does.
Like you see that italicized Latin text, and it immediately feels like a fundamental law of physics. It feels tidy. It feels resolved.
But what if I told you that the naming system giving us these authoritative labels is actually a chaotic mix of posthumous honors, medical compromises, and giant legal loopholes?
It is a remarkably messy human endeavor, and honestly a highly dramatic one.
Yeah, I had no idea until I started reading the material for today.
The story of how we label the microscopic world is full of heated rivalries, impossible compromises, and some surprisingly fragile rules.
So today's deep dive takes a massive stack of taxonomic research, history, and microbiology notes to explore the surprisingly wild world of binomial nomenclature.
Which is a mouthful.
It is, but specifically, we are going to look at how microbes actually get their names and why those names are constantly threatening to fall apart.
To set the baseline for you, when we say binomial nomenclature, we are talking about a two-part Latinized scientific name.
Right, we all know the drill from high school biology.
Hopefully, yeah.
You have a capitalized genus, which groups related organisms together, followed by a lowercase species epithet to distinguish the specific organism.
And you throw both parts in italics to look official.
Like Staphylococcus aureus.
Exactly.
And a quirk you might have noticed is that after the first time it gets written out in a document, that genus name gets abbreviated to just a single letter.
Which saves a lot of typing.
Oh, totally. Which is why you constantly see things like E. coli popping up in those food recall notices instead of the full formal name.
Right. And the structure itself seems perfectly logical on the surface. But the history of how we arrived at this system and how we eventually had to, force microbes into it is anything but tidy.
Okay, let's unpack this. Because to understand how microbes get their names today, we really have to look at the massive communication crisis that forced scientists to invent this system in the first place.
Yeah, if you rewind the clock to before this two-part system existed, scientists were using what we call polynomial names.
Poly meaning many?
Right. These were chaotic strings of Latin descriptive phrases that could be up to a dozen words long.
Wait, a dozen words for one name?
I know. Instead of a simple name, you essentially got a tiny, incredibly dense paragraph describing the organism's leaves, its color, its habitat, and its smell.
I mean, I can't even imagine trying to write a medical paper where every time you mention a bug, you have to write a 12-word Latin paragraph just to ensure everyone is on the same page.
It was exhausting. But the real nightmare was that these long, rambling phrases changed depending on which author was writing about the organism.
Oh, of course they did.
Yeah, one scientist in France might focus on a plant's flowers, while a scientist in England focused on its roots.
So they'd have completely different 12-word names for the exact same species.
Exactly. It made cross-referencing specimens across Europe practically impossible. I mean, the scientific community was drowning in duplicate, conflicting descriptions.
So who finally stepped in and stopped the madness?
Carl Linnaeus solved this communication crisis in the 1750s.
Oh, right, Linnaeus.
Yeah, he published Species Plantarum in 1753, and then the 10th edition of Systema Naturae in 1758. And he introduced these short, stable, two-word tags for plants and animals.
Which was revolutionary.
Completely. Suddenly, everyone had a universal filing system.
But looking at the notes for our deep dive today, there's a glaring omission here. Linnaeus didn't classify microbes at all.
He couldn't?
Why not?
Well, Anthony van Leeuwenhoek had only just seen what he called animalcules under his rudimentary microscopes in the 1670s.
Animalcules, what a great word.
Right. He was peering through these tiny, handcrafted glass beads and seeing these little entities swimming around, but nobody had a framework for organizing this invisible world yet.
The technology just wasn't there.
Exactly. It actually wasn't until the 1870s that a German botanist named Ferdinand Kohn became the father of bacterial systematics.
So he brought Linnaeus' system to the microbes.
He did. He essentially ported Linnaeus' plant and animal system over to bacteria, sorting them into genera based primarily on their visual shape and their behavior.
Which sounds like a brilliant leap forward, but my understanding is that it actually led to a century of completely chaotic, duplicated, and unverifiable naming in the microbiological world.
It was an absolute mess.
Like you had scientists discovering things left and right, throwing names at them based on whatever microscopic shape they saw.
Yeah, and creating a tangled pile of data that makes the old polynomial days look organized.
Wow.
The situation reached a breaking point. The scientific literature was so polluted with bad data, redundant names, and poorly described microbes that they had to take drastic action.
The way they finally solved this is absolutely wild to me.
It's pretty extreme.
It's the equivalent of a country declaring all pre-1980 property deeds void unless you refiled your paperwork with the new central government.
That is a perfect analogy.
Yeah.
Because they executed a literal legal reset for biology.
I still can't believe they actually did this.
Yeah. In 1980, three taxonomists, Skerman, McGowan, and Sneath, they published a document called the Approved Lists of Bacterial Names.
Okay.
And the governing code for this whole system declared that January 1st, 1980 was the new day one of bacterial nomenclature.
Wait, hold on. They just deleted 2 centuries of scientific literature overnight.
Essentially, yes.
How did scientists not riot in the streets? If a name wasn't on that specific list, you're saying it just legally ceased to exist.
Erased. Completely erased. It did not matter how old the name was, how famous the researcher who discovered it was, or how many papers it had been cited in.
That is brutal.
If it was not on the 1980 approved list or validly published after that date according to the new strict rules, it had no standing in nomenclature.
Wow.
The scientific community basically agreed to swallow this bitter pill because the alternative, continuing to dig through 200 years of conflicting, poorly documented names, was threatening to stall microbiological research entirely.
That is just staggering. It's a complete institutional reboot. And while we are talking about drastically redrawing the maps of life around this time, we have to talk about Carl Woese.
Oh, absolutely.
Because in 1977, right on the eve of this legal reset, he fundamentally changed how we even categorize these organisms.
He did. Woese utilized a brand new technique, ribosomal RNA sequencing.
Okay, so looking at the genetics.
Right.
Yeah.
Instead of just looking at the shape of a microbe under a microscope like Ferdinand Kohn did, Woese looked at the factory machinery of the cell itself.
Ribosomal RNA.
Exactly. Ribosomal RNA is highly conserved, meaning it mutates very slowly over billions of years.
Yep.
By comparing the genetic code of this RNA across different organisms, he proved that life doesn't just split into 2 basic groups of prokaryotes and eukaryotes like everyone assumed.
Right. He proved it actually has 3 domains, bacteria, archaea, and eukarya.
Which forced a massive renaming project above the genus level. For an entire century, archaea had been misclassified.
Because they looked like bacteria?
Exactly. Because they looked like bacteria under microscope, scientists just assumed they were unusual bacteria.
Thought they weren't.
No. Woese's molecular evidence revealed that, genetically speaking, archaea are as different from bacteria as you and I are from bacteria.
Yeah, it's crazy.
That singular discovery forced taxonomists to completely reorganize the domain-level map, which is why archaea even exists as a taxonomic category for you to learn about today.
So we have this incredibly strict legal framework established, complete with the day one reset and a brand new genetic map of the domains of life. But with all those sweeping rules in place, how does a single newly discovered microbe actually earn its specific 2-word tag?
To understand that, we need to look at how these names function as literal records of human history.
Let's start with maybe the most famous one in the notes, Escherichia coli.
Good old E. coli.
Right. This organism was isolated back in 1885 by a German pediatrician named Theodor Escherich, who was actually trying to figure out the cause of infant diarrhea.
But he didn't name it after himself.
No, he originally called it Bacterium coli commune.
Right, he was focused on the location and the function. It wasn't until decades later, in 1919, that researchers Castellani and Chalmers created the genus Escherichia posthumously.
Specifically to honor him.
Yes. And the species epithet, coli, simply denotes its habitat, the colon.
Good, makes sense.
This case perfectly illustrates how species epithets are often descriptive, detailing the bug's habitat or appearance, while the genus might be an honorific formed from a scientist's surname.
So Escherich got his name peacefully, years after the fact. But looking at these notes, what happens when multiple people claim a discovery at the exact same time? I mean, naming becomes a literal battlefield.
It absolutely does. The bacterium responsible for the Black Death is a fantastic example of names moving around to settle historical scores.
Ah, this story is wild.
Yeah, the plague bacterium was originally named Pasteurella pestis in 1896 by researchers working in Louis Pasteur's scientific orbit.
But the name today is Yersinia pestis. How did Pasteur lose the title?
Well, as biochemical and genetic analysis improved by 1944, researchers realized this bacterium was distinct enough from the rest of the Pasteurella family that it deserved its own genus entirely.
Okay.
And this provided an opportunity to correct the historical record. Alexander Yersin was a Swiss-French bacteriologist who co-discovered the plague bacillus in Hong Kong in 1894.
Ah, so he was there first.
And what's amazing is the context. He discovered it while working in a makeshift lab in a literal high-stakes race against the famous Japanese bacteriologist Kitasato Shibasaburo during a massive outbreak.
So when they created the new genus in 1944, they named it Yersinia to cement his victory in that specific race. The name itself is a trophy.
Precisely. It is a historical marker.
But sometimes a name isn't a trophy for a scientist. Sometimes it's a memorial for a tragedy.
Yeah, that's true too.
I was looking through the notes on an outbreak from July 1976 that really stood out. At an American Legion convention in Philadelphia, 182 attendees suddenly developed this mysterious, incredibly severe pneumonia.
And 29 people died.
Right, a completely terrifying scenario, especially because the culprit was entirely invisible to standard medical techniques.
The CDC, the Centers for Disease Control and Prevention, launched a massive investigation, and it took them 6 straight months to finally culture the hidden culprit.
6 months?
Yeah. It simply refused to grow on the standard agar plates they used in the lab. It needed highly specific, unusual nutrients to survive outside the human body, which is how it had completely escaped detection for so long.
So when they finally did manage to coax it into growing in a culture in 1977, their genetic and biochemical tests showed it was so incredibly unique, so functionally different from every known bacterial family, that scientists had to create an entirely new genus for it.
And they named it Legionella pneumophila.
It acts exactly like a memorial plaque for that exact outbreak.
It does.
The genus is permanently named after the American Legion Convention, and pneumophila literally translates to lung-loving.
Yeah, it's very descriptive.
Whenever a doctor writes that name down, they are documenting a piece of 1976 history.
That historical weight is profound. But there's a mechanical layer to this naming process that governs all of these examples, and it's arguably the most rigorous and strange rule in the International Code of Nomenclature of Prokaryotes.
Or the ICNP.
Right.
Oh, the freezer tube rule. This absolutely blew my mind when I read it.
It's wild, right?
According to the ICNP, every single validly published bacterial or archaeal name must be legally tethered to what they call a type stream.
A physical, living, biological reference specimen. A name cannot just be an idea in a textbook.
The rules state this type strain has to be deposited in at least 2 internationally recognized culture collections in different countries.
Right. For redundancy.
Like the ATCC in America, which is the American Type Culture Collection, basically a massive, highly secure warehouse of frozen biological samples, or the DSMZ in Germany.
Exactly.
But wait, let me get this straight. If a name is legally tethered to a physical sample, if someone unplugs that specific freezer and the liquid nitrogen runs out and those physical cells die, does the name just become unusable?
That is a very real fundamental vulnerability in the system.
You're kidding
No,
me.
the entire nomenclature framework assumes physical culture continuity. The name is anchored to an actual glass tube of cells sitting in a deep freeze.
That's so stressful.
If that type strain is lost and cannot be recovered from any official collection globally, the scientific name itself can essentially become void.
That is wild. Tying a concept to a physical freezing vial sounds incredibly rigorous, but reading further into the notes, it seems like it creates 2 massive scientific headaches for modern microbiologists.
It really does.
Which brings us to the biggest debate of all. What even is a microbial species?
This raises an incredibly important question about how we classify life. You've probably heard of the classical biological species concept.
Ernst Mayer's idea, right?
Yes, Ernst Mayer's famous idea that a species is a group of organisms that can interbreed with one another and produce fertile offspring.
That makes perfect sense for birds or beetles or elephants.
It works beautifully for them. But bacteria and archaea reproduce asexually. They just divide.
Right.
And to make things infinitely worse for taxonomists, they constantly swap genes across what look like established species lines through a process called horizontal gene transfer.
So instead of inheriting traits vertically from a parent, they're just swapping genetic trading cards with their neighbors in the environment. Imagine if you could just bump into someone on the street and suddenly acquire their eye color or their ability to digest milk.
That is exactly what bacteria do. They share antibiotic resistance genes and metabolic tools across completely different genera.
Wow.
So if they don't interbreed and they constantly share DNA across boundaries, how do you define a distinct bacterial species?
The notes say modern taxonomists have had to quietly redefine the concept of a species as an operational statistical threshold.
Yes, they use something called average nucleotide identity, or ANI.
How does that work?
They look at the complete genetic instruction manual of two bacteria. You line up millions of letters of genetic code side by side. Most commonly, taxonomists agree that two strains belong to the same species if their whole genomes share roughly a 95 to 96% identity match.
So it's basically like grading on a curve.
Kind of, yeah.
If you get a 96% match on the DNA test, you get to be in the club and keep the name, but a 94% means you're an entirely different species. That is just a human-drawn line on a continuous genetic landscape.
Taxonomists openly debate this all the time. Are bacterial species real biological units found in nature, or are they just convenient administrative fictions we invented so we can organize our databases?
And the absolute clearest example of this tension is the massive debate between E. coli and Shigella.
Oh, this is a classic.
If you are a doctor listening to this right now, or if you've ever had a severe case of food poisoning, you know Shigella causes severe, sometimes deadly bacterial dysentery.
It's awful.
But genomically speaking, several species of Shigella are so remarkably similar to E. coli that they sit comfortably well within that 96% ANI range.
Which means, technically, under the rules of genetics, they should be classified as the exact same species. Shigella should just be considered a particularly nasty strain of E. coli.
But they don't change the name.
What's fascinating here is that taxonomists flat out refuse to merge them. They intentionally keep them separate, purely for medical utility.
Because of the doctors.
Right. Think about the real-world stakes. Clinicians, public health surveillance systems, and over a century of medical literature depend on the name Shigella, meaning the dangerous dysentery bug.
Yeah, that makes sense.
If they merged it into the massive E. coli family, it would throw hospitals into absolute clinical chaos. Doctors would be confused about what exactly they were treating.
So the taxonomists consciously sacrifice scientific phylogenetic purity to prioritize the safety of clinicians and patients.
Exactly.
It's a pragmatic lie. We keep the inaccurate filing system because the biological truth is too dangerous for the hospital floor.
But remember that rule we discussed? The requirement for a physical culture in a freezer tube.
Oh, right. The ICMP rule.
That creates an even bigger headache known as the Great Plate Count Anomaly.
Because the legal code explicitly requires a physical cultured specimen to validate a name, we have a massive problem with the 99% of environmental bacteria and archaea that simply cannot be grown in pure culture in a lab.
We can't grow them, but we know they exist.
Because we've detected them by scooping up ocean water or soil and running pure environmental DNA sequencing.
We can see their genetic ghosts, we know exactly what their DNA looks like, and we know they play massive roles in global ecosystems.
But because we can't figure out the exact magical mixture of nutrients and pressure to grow them in a petri dish so we can stick them in a freezer tube, technically, the vast majority of microbial diversity on Earth cannot be formally named.
It's a huge blind spot. The workaround taxonomists developed for this is the provisional candidatus status.
Candidatus, like a candidate.
Right. If you have the genome mapped, but you don't have the physical culture in hand, you park the organism in this naming limbo.
The most famous example from the notes being Candidatus pelagibacter ubique.
Discovered by Stephen Giovannoni's group, it is an extremely abundant marine bacterium in this Air 11 clade.
Huge for the oceans, right?
It is massively important to the ocean's carbon cycle. But despite its global importance, it sat in that provisional candidatus status for years.
Because they couldn't grow it.
Exactly. It wasn't until a culturable representative was eventually painstakingly isolated that the name could be properly validated.
Okay, let's unpack this. We've seen how prokaryotes, bacteria, and archaea are strictly governed by this Linnaean sister, the ICNP, with its liquid nitrogen freezers and its 95%
It's
thresholds.
a very rigid system.
But there is a massive exception to all of these rules, an exception that you, listening right now, have intimately lived through recently: viruses.
Viruses completely break the mold. They are governed by an entirely different non-Linnaean code run by the International Committee on Taxonomy of Viruses, or the ICTV.
Here's where it gets really interesting. Think back to 2019. The virus causing the global pandemic was not given a traditional binomial genus species name like Legionella pneumophila.
No, it wasn't.
The ICTV officially named the virus SARS-CoV-2, but the World Health Organization separately named the disease it causes COVID-19.
That separation was not an accident. It was a highly deliberate public health strategy.
How so?
The WHO has best practice guidelines, established in 2015, that explicitly ban naming new human pathogens, or the diseases they cause, after geographic locations, animals, or ethnic groups.
Because of the massive economic and social damage caused by our careless naming in previous outbreaks.
Exactly.
Things like swine flu devastating the pork industry, or the stigma attached to Middle East respiratory syndrome.
By intentionally decoupling the virus name from the disease name, they aim to avoid those stigmas.
Even if it confused people.
Right. The result was genuinely confusing for the public. You had media and politicians using coronavirus, SARS-CoV-2, and COVID-19 interchangeably, even though they technically referred to a broad virus family, a specific virus particle, and the resulting human disease, respectively.
It was a lot to keep straight.
But it was deemed a necessary separation to prevent the kind of geographical targeting we've seen historically.
And even when the scientific reasoning for a name change is perfectly clear, human habit is incredibly hard to break. Look at what happened with C. diff. In 2016, genomic evidence forced a major reclassification. Scientists mapped its DNA and realized its underlying genetic blueprint was wildly different from the rest of the Clostridium family.
It was an imposter.
So they had to kick it out of the family tree and officially rename it Clostridioides difficile.
Scientifically, genetically, it was absolutely the right move to make.
But hospitals, textbooks, and even peer-reviewed medical journals just kept using the old name for years.
Because of habit.
Why? Because the abbreviation C. diff was so deeply, thoroughly entrenched in the daily culture of medicine, the humans simply lagged behind the taxonomy. They didn't want to learn a new word when the old one worked fine on the chart.
It perfectly illustrates how incredibly slowly these nomenclature updates propagate into actual clinical practice, regardless of how formally correct they are.
So what does this all mean? Why does all of this complicated taxonomy, these debates over 95% thresholds, and these freezer tubes actually matter to you?
Because this naming system is the load-bearing infrastructure for all of global public health.
It connects everything.
You cannot track antibiotic resistance across continents and decades without it. Take MRSA, methicillin-resistant Staphylococcus aureus.
Right.
The only reason we can track that specific threat as it moves from a hospital in London to a clinic in Tokyo is because the binomial name anchors our global surveillance databases to one agreed-upon identity.
It's the exact same principle for managing food safety recalls when Salmonella enterica gets into a batch of lettuce, or when pharmaceutical companies are developing new vaccines.
Exactly.
The entire modern medical system rests on the fundamental assumption that a scientific name points to one stable, internationally agreed-upon bug.
Without that, the whole system collapses.
Let's summarize the journey we've been on today. We started with the basic assumption that scientific names are permanent, static truths carved in stone.
And we saw that's definitely not the case.
But the reality is they are living, breathing human attempts to draw lines on a chaotic genetic landscape. It requires legal resets that wipe out 2 centuries of history.
And tubes of frozen cells in high-security vaults.
Right. Provisional waiting rooms for uncultured DNA, and sometimes consciously sacrificing scientific purity so that doctors on the ward know exactly what disease they are fighting.
If we connect this to the bigger picture, it really highlights a profound tension between nature's complexity and human administration.
Absolutely.
We just learned that bacterial species are largely human-drawn statistical fictions based on a 95% grading curve, created purely for our own administrative convenience.
Which is still hard to wrap my head around.
So I want to leave you with this thought to ponder. If that is true, as we push further into the microscopic world with increasingly advanced genetics, Are our medical treatments actually tailored to the natural reality of the microbes themselves, or are we just treating our own filing systems?
That is a wild thought to end on. Thank you for joining us on this deep dive. Hopefully you'll think twice the next time you see an italicized microbe name flashing across a warning label. Until next time.